and thank you all for having me here. I'm sorry for the technical difficulties on my end, and I was hoping to have my webcam sharing right now, but I'm sorry that I wasn't able to get that up and running. I'm Mike Gomez, and I'm an assistant professor at the University of Washington, and today I'm going to be talking about the use of microorganisms, which can complete mineral precipitation processes for the mitigation of earthquake-induced soil liquefaction. Specifically, as we get started here, I wanted to introduce what's referred to as biogeotechnics, and it's a relatively new field. And so in brief, biogeotechnics is the study and utilization of biological, chemical processes, reactions, and mechanisms found in nature to solve engineering problems related to Earth systems. And there's really two different flavors of these biogeotechnical solutions. One is referred to as bio-mediated. That's where we're using microorganisms or some sort of other biological process directly to do something of interest. And the other one is bio-inspired, where maybe we're looking at how ants tunnel or how worms burrow, and we're trying to extract principles that can be applied to a geotechnical system and solve problems that way. And so really the whole premise is that natural systems have persisted and obviously adapted over geologic time. And so potentially they can offer us some insights with respect to designing and building civil systems more efficiently, restoring and remediating geosystems, and enabling the discovery of new technologies. And I think it's a really exciting time right now of discovery in this research community. In the United States, the National Science Foundation, of course, has sponsored what's referred to as the CBVG, or the Center for Biomediated and Bio-inspired Geotechnics. It's a center that's devoted towards looking at these types of technologies. There's many other centers like this throughout the world. And I think it's a really exciting time to look and see the potential that's right here in the soil to do all of these processes and more efficiently and in more environmentally friendly ways. So when we begin here, here's just kind of the overall outline of the talk. I'll first provide an introduction to what MICP is. Then we'll talk a little bit more about the engineering behavior of MICP through some element testing. We'll talk then about some process upscaling experiments that we've done to try to look at how we can deploy this type of process at field scale. And then some of the more recent experiments which we've done looking at, in particular, permanence aspects and understanding sort of the mineral precipitation process more fundamentally. And then we'll end with some conclusions and, of course, questions. Okay, so really overall, the motivation for microbial-induced calcite precipitation, or really any bio-mediated geotechnical process, is the idea that we have traditional ground improvement processes which use high mechanical energy and energy-intensive materials. They're extremely effective. However, they come at significant environmental cost. We're talking about something on the order of 40,000 projects or $6 billion per year. And of course, these processes are resulting in things like greenhouse gas emissions, aqueous toxicity. And so the idea here is really to look to alternative technologies, which can potentially enable similar engineering improvements with reductions in environmental impacts. And so biomediated soil improvement technologies really fill this gap by leveraging these natural biological and chemical reactions in the subsurface to instead sort of replace some of these more traditional technologies. In terms of reaction network of how MICP works, it's a biomediated cementation process that can really transform loose granular soils into stiffer, stronger rock-like geomaterials. It does so through a series of reactions, the first of which is urea hydrolysis, wherein urea is actually being hydrolyzed by a microorganism into ammonia and carbonic acid. This is then, as the ammonia enters in free solution, pulls a proton off of water, becoming ammonium and producing hydroxide. This hydroxide ion is important because it's going to increase the pH of surrounding solution. And as the pH increases, this carbonic acid, which is produced in the first step, is going to become increasingly bicarbonate and carbonate. And when there's sufficient carbonate available and we add calcium salts into the treatment solutions, we're able to get calcium carbonate precipitation both on particle surfaces and at particle contacts. The result really is that we see large increases in initial shear stiffness, shear strength, and cyclic shear resistance of these materials with relatively minor reductions in porosity and hydraulic conductivity. Largely, the pores are going to remain open and conducive to flow. And so this process has been researched extensively for almost 2 decades now. Some of the engineering applications include general ground improvement, liquefaction mitigation, concrete remediation, wherein you might be able to seal up cracked materials, slope stability and erosion control, contaminant mobilization, where they're actually trying to co-precipitate out some other contaminant, which may be divalent like strontium, and so you're incorporating it in the mineral itself. And then the construction material is where maybe you don't have access to timber or Portland cement, and so maybe this could be a viable process in some of those locations. Really, our focus has been on strictly liquefaction mitigation, and I'm excited to kind of talk through both the process and some of the results related to that today. In terms of the treatment approaches, it really can be done in a variety of different ways. Sort of the traditional approach is what's referred to as bio-augmentation. That's the introduction of non-native microorganisms with desired metabolic or enzymatic capabilities. Specifically here, we apply Sporocycina pasteurii most commonly. Of course, this microbe has the ability to hydrolyze urea. That's the capacity that we need here. But there are issues with things like spatial uniformity, overall cost competitiveness, and environmental impacts. Instead, more recently, what we've been looking at is what's referred to as biostimulation, wherein really all we're doing is injecting substrates and changing the environmental conditions to enrich for specific native microbes of interest. In this case, we're enriching the native community specifically for those that can complete ureolysis. And so this has been more recently explored for field scale applications. Of course, it has the potential to improve the spatial uniformity and ease of permitting of this type of technology because we're not introducing exogenous bugs. And there's significant reductions potentially in treatment costs and material and energy requirements of something like this. And recent work really suggests that these indigenous urolytic bacteria are ubiquitous in many different soil types. And so it's a capacity there that all we have to do is really learn how to enrich for. In terms of mechanisms, from a more mechanical standpoint of how this cementation is going to improve soil behaviors, there's a variety of mechanisms at play here. The first of which, of course, is interparticle bonds. It's shown in the schematic there to some extent. And the other piece of it is also the coating of soil particle surfaces, which is going to change angularity. And of course, the addition of mineral solids itself is going to induce some densification to the soil matrix. Of course, when we shear these types of material, we see increases in shear stiffness and strength, and really those attributes are coming from a variety of different mechanisms, some of which are mobilized at smaller or larger strains respectively. Of course, the cohesive calcite particle-to-particle bonds, those are relatively stiff and strong at small strains. However, once those fracture, we're actually seeing sort of increases in material dilatancy due to increases in bulk density and particle angularity. And so there's a variety really of mechanisms at play here, both at the small and larger strains during failure. We can also use various process monitoring techniques. As one example, shear wave velocity is commonly used to track cementation progression. What we're showing here is what would be voltage versus time lag. And so these are our Bender element measurements that we're actually picking up from a column. As you can see, the frequency of the wave, the amplitude, and actually the arrival time of that wave is increasing as we progress through cementation, really moving ourselves from something that would be 150 meters per second to something closer towards 1,000 meters per second, let's say in a period of 7 days or so. We can kind of think of this as actually moving these materials through geologic time, whereas we're taking something that's initially more or less a Holocene-like material, and within a day, we're able to see a appreciable change in shear wave velocity. And by day 7, we're really approaching a condition that's more or less like soft rock. And so this process allows us really to improve these materials dramatically. In terms of sort of the biogeochemical side of things, of course, I mentioned the variety of chemical precipitation and ureolysis reactions that are occurring here. What we see for solution pH versus time really is initially we sort of have two different stages. We have a stimulation stage and we have a cementation stage, and we can look at both solution pH versus time or urea degradation versus time. During stimulation, the objective here is to provide nutrients and to provide urea to actually enrich the native microbial population. And the result is the following. When we apply urea, the pH goes up, indicating that we have ureolysis, and we can see the same thing here in terms of urea degradation. And once we move into cementation, now we're actually applying calcium salts, and we see that we never really get to the same pH level, and we oscillate back and forth. And really, this is the result of carbonate buffering reactions and essentially indicating that we're now precipitating out calcite. As you'll see here in terms of urea degradation, once we get past a certain critical point, really the microbial population is converging to the point that we're able to hydrolyze the injected urea practically every day that we apply it. We take this information, of course, looking at the reaction network, and we're able to deduce something by how quickly the urea degrades. Of course, this is a sort of a slope that's dependent on a variety of things. Some things are related to the enzymatic parameters, that is a maximum rate and a half saturation coefficient. Those are properties of the enzyme themselves, which sort of reflect their concentration dependence and speed. The urea concentration, of course, which we apply, and X, which is actually the urolytic cells. So by looking at this from this standpoint, we're actually able to deduce how efficient our enrichment is and how successful we are at targeting the specific microbes of interest. Okay. So that's kind of the basics of MICP, and now I kind of want to move into talking a little bit about some of the engineering behavior tests that we look at. The first of which is our sort of standard soil column experiments. Really the objective here is to examine changes in engineering properties as a function of a variety of things, including treatment techniques, soil material types, biological differences, and other site conditions. And so this is sort of a venue that we can test out sort of different treatment formulations and understand how this might impact engineering properties and the biogeochemical process. Of course, in these types of tests, what we're doing here is we're monitoring aqueous chemistry by taking samples at mid-height, oftentimes in some of our experiments. We're actually looking at shear wave velocity changes as well using Bender element sensors. And so we're cleaning a variety of different measurements to tell us both about kind of real-time changes in the mechanical attributes as well as the biogeochemistry. In terms of soil column experiments that we've done in the past, we've looked at an extensive range of different alluvial sands and from different environments here, fines contents of less than 13% or so. By and large, what we find is that these tests suggest that these native microorganisms exist in a broad range of natural materials, independent of anything related to grain size or depositional environment. We do see changes in what we would refer to as microbial lag time, that is the time needed to stimulate them and get them up to a point where we can transition to cementation. But by and large, these microorganisms are ubiquitous, and they're existing in these different locations and just ready to go in terms of being able to complete this process. We've been able to now summarize kind of a range of post-treatment engineering properties obtainable, including changes in Ds, unconfined compressive strength, and permeability. When we look at the data set that we have from a variety of these soil column experiments, and we bin them by fines content, in the light gray being those cleaner materials and the darker gray being the ones with higher fines, as well as particle size, what we see is by and large consistent trends in unconfined compressive strength with some modest dependence on fines content. But what we're really talking about is moving something that initially is uncemented sand at about 2% calcite content. It's able to stand up on its own and complete an unconfined compressive strength test. And then we have a spectrum of improvement all the way up to something like 12% calcite content, where we have an unconfined compressive strength of 5 MPa, or basically weak rock. We can also look at VS, or shear wave velocity changes, as a function of calcite content. We really don't see too much dependency on fines content overall. And we're really thinking about something that's closer to 1,500 meters per second at about a calcite content of something like 10%. We've also done some select measurements in terms of permeability, mostly on clean sands. What they generally reflect is that we're talking about an order or so, even at the high end of cementation. So permeability really isn't reducing all that much, unless we get some really siltier or finer grain materials at which we have observed more significant changes. Moving forward to talk a little bit more about some of the more recent testing we've done, we've really focused on direct simple shear testing to specifically examine the undrained cyclic response of these sands for understanding the liquefaction behavior of these materials. Of course, what we're trying to do in these different tests is evaluate liquefaction triggering and the consequences of triggering as a function of a variety of different variables, including cyclic demand, initial and final relative density, cementation magnitude as reflected by either a calcite content or a shear wave velocity, stress history, and material damage. And of course, these tests are really complementing a larger effort here, which is ongoing through CBBG, including centrifuge testing and some numerical modeling efforts to try to capture these behaviors for field scale deployment. In terms of our specific lab tests, of course, we've seen that MICP can improve the undrained monotonic and cyclic behavior of sands through a variety of different tests. And despite these recent high-quality tests on a variety of soils, really the effect of low levels of cementation have been relatively poorly characterized. And so our focus really has been on the transition from the uncemented to cemented material, really to guide field implementation and understand what that effect of cementation is on these different behaviors, and to minimize environmental impacts and costs, which of course scale with the amount of cementation that we need at a given project. The physical properties of MICP also might provide for an extension of our results, and sort of an analog to the behavior of naturally cemented deposits, which are found throughout the world, and oftentimes are actually cemented with very similar minerals to calcium carbonates. And so we feel like our results are able to sort of circumvent the challenges related to sampling and disturbance of actually getting materials that were naturally cemented from the field. In terms of specimen preparation for these particular tests, we dry pluviated to a relative density of about 30%. We then augmented, and where we're injecting cells and applying cementation treatments to achieve varying changes in shear wave velocities, specifically looking at 0 to 100 meters per second in terms of changes. We also had uncemented control tests, and this was really to compare the effect of cementation to the effect of densification and to provide some baseline for our understanding of sort of how those behaviors are evolving. And then the specific undrained shearing event that we applied was a range of CSRs anywhere from 0.1 to 0.3. The shearing was completed until we achieved 24% double amplitude shear strain. Afterwards, we reconsolidated all of these and measured reconsolidation vertical strains after those shearing events. The other measurements that we were completing were also shear wave velocity measurements at different points in time to try to track cementation degradation and damage. And then we also completed soil calcite contents post-testing, as well as looked at the effect of cementation on dry density. And here we're really trying to say that the cementation that's being added to these samples is also going to change sort of the relative density, one could think, in a way that may be significant at higher levels of cementation and really maybe not so significant when we're at this lower levels that we looked at. So in terms of our specimens in particular, what we were able to obtain again is VS anywhere from 0 to 100 meters per second. That's what I'm plotting here on the y-axis is the change in VS from cementation. We've also got two x-axis axes here. One is calcite content on the first one, and the other one is change in relative density from cementation, where basically we're saying that the cementation occupies some volume, of course, and using the density of calcite, we can say approximately what the relative density change would have been if that cementation were solid soil minerals. What we find overall is that the relative density resulting from a cementation increase is relatively small. We're talking about basically 5.5%, even at the high end of the cemented spectrum that we looked at. And the calcite contents are really quite small that we looked at, anywhere from 0 to 0.9% by mass. As you can see here, these plots really show a strong correlation between change in shear wave velocity and calcite content, which really reflects sort of consistency in terms of the fabric that we were able to induce with the cementation processes that we used. In terms of what these materials actually look like, here's an image of both the uncemented specimen as well as sort of a specimen at our higher end of change in shear wave velocity of 77 meters per second. They're both at 200 magnification here, and you can kind of see it's really kind of hard to distinguish the difference between the two. There's not a ton of cementation here. We can zoom in a little bit more, and what you really find is, yes, there are certainly calcite crystals present on some of these surfaces, but by and large, this cementation is more or less a dusting, right? It's not really the cemented materials that we see in some of our other tests where we're going to 1500 meters per second, right? This is a single treatment and relatively low cementation overall. In terms of engineering behavior and the cyclic test that we did, we got a spectrum of behaviors, of course, depending on the amount of cementation present. Here, what we're showing is some test results for a CSR of 0.2, an initial relative density of about 30%. This is the untreated response, both in terms of shear stress versus vertical effective stress, shear stress versus shear strain, and RU, or pore pressure ratio, versus cycle number. As you can see here, we're essentially triggering liquefaction if we look at it in terms of an RU of 0.95 within about a half a cycle or so. In terms of shear strain, we're also triggering liquefaction in less than a cycle. And when we look at essentially the response, everything is failing almost monotonically. As we progress forward, however, this is now a change in shear wave velocity of about 54 meters per second. So this is about middle of the road in terms of our spectrum. You can see that we're able to tolerate many more cycles prior to liquefaction triggering. And when we trigger liquefaction, we're at something like 12 cycles or so. Afterwards, however, you see that the strains seem to accumulate relatively rapidly after triggering does occur. Once we move forward and now we're at a shear wave velocity increase of 95 meters per second, what we can see again is many more cycles required to get that same amount of contraction. Once we do fail though, the strain accumulation piece doesn't look too much more different than what we saw at 54 meters per second, but our needed cycle numbers to actually trigger liquefaction in this material is approaching something like 650 cycles. And so we see a significant change really in the pre-triggering behavior. And when we look at the strain accumulation post-triggering, it's really not all that different, which is not to be super surprising considering that the densification that we provided is relatively small. This is a small amount of cementation. When we look at pre-triggering behaviors, of course, what we can see is now I'm plotting cycles to 3% single amplitude shear strain. This was our selected liquefaction triggering criterion. And we're plotting it versus delta Vs from cementation. Again, it's a spectrum from 0 to 100 meters per second. And as you can see, there's a wide variety in the number of cycles required to trigger liquefaction. Some of them are uncemented points. Those are in the circles at 0. And our cemented points are the ones in the triangles. And you can see with the CSR, we're basically ramping up and changing cyclic demand. What we end up seeing is regardless of CSR, we're seeing about anywhere between 2 to 4 orders of improvement in terms of the behavior or number of cycles required to trigger with no cementation versus the number of cycles required to trigger liquefaction for 100 meters per second sample. We also see better agreement on the basis of delta Vs. If we plot the same relationship with calcite content, we actually see that the number of cycles to liquefaction triggering correlates very well with a change in a small strain shear stiffness, but not necessarily with calcite content, which was an interesting finding and really suggests that the triggering behavior might be more strongly influenced by the bonding between particles. When we plot that same graph now versus cycles to 3% single amplitude shear strain versus total relative density, now we've got many more uncemented specimens. These are the ones that we prepared at different relative densities. What we can see is that there's significant improvement certainly with densification, but the improvement that's afforded by cementation, which is basically just a vertical line suggesting that there's almost no change in relative density and that it's just increasing straight up in terms of number of cycles required, really suggests that the cementation is providing benefits in terms of liquefaction triggering that really can't be practically obtainable through densification alone. As we keep going here, we were also taking this same plot, which is now cycles to 3% single amplitude shear strain and delta Vs, and we use them to develop liquefaction resistance curves. As you can see in the previous slide here, really we don't have a lot of tests at the exact same cementation level. And so what we did is we instead used the relationships that we developed between delta Vs and cycles to 3% single amplitude shear strain. We then picked off various thresholds of delta Vs, 10, 25, 50, 75, and 100, and we used them essentially to develop our liquefaction resistance curves. When we did that, it looked like the following, which is CSR versus number of cycles to 3% single amplitude shear strain. What you can see here is that each of these curves represents a different cementation level. Going from the light end of the spectrum, this is our uncemented condition, all the way up to 100 meters per second in the dark red. Interestingly, what we found is that we had greater improvements in these behaviors really at the lower CSRs at 0.1 as opposed to up to 0.3, really suggesting that at these lower cyclic demands, we might be having more of a fatigue-like mechanism occurring rather than an immediate yielding of the material, which is occurring likely at higher CSRs. We can also plot this in a different way here, which is just to divide these to look at an improvement ratio. And what we end up finding again is that we're talking about 2 to 4 orders increase in the number of cycles to trigger liquefaction, but it is a spectrum, right? There is a difference between a higher loading demand versus a lower, and really the lower is where we're seeing the maximum benefit. In terms of other criteria that we looked at, we also then plotted for all of our specimens the number of cycles to 3% single amplitude shear strain, as well as cycles to an RU of 0.95. Both are commonly used as a liquefaction triggering criterion, and we wanted to see if there was any bias in sort of the triggering criterion that we used. Overall, they're pretty much following the one-to-one line, except at really low cycles to single amplitude shear strain or high cyclic demands in the uncemented conditions. But by and large, really either liquefaction triggering criterion is giving us basically consistent results. We weren't really satisfied with that, and so we looked a little bit more at the pore pressure generation behavior. And so even though these liquefaction triggering criterions were very similar, we wanted to investigate this a bit further. And so we looked at actually two different intervals here to better understand the effect of cementation on pore pressure generation. We looked at the first half, which was essentially an RU of 0 to 0.475, and then the second half of the pre-triggering pore pressure generation, which would be basically the number of cycles required to change the RU from 0.475 to 0.95. And so this was a little bit unconventional. What we were trying to do is break up the pre-triggering behavior into two different segments of equal pore pressure generation. And so what we expected to see is that the second half of pore pressure generation, of course, would occur in less cycles because there'd be higher pore pressures, lower effective stresses, and regardless of cementation, the material should soften and require less cycles for that second half. However, we weren't entirely sure how those differences might look when we were comparing sort of our cemented samples to our uncemented samples. When we plotted the first half, now I'm showing cycles from RU of 0 to 0.475 versus delta VS versus cementation. We get something that looks very similar to the previous plots where we were plotting cycles to 3% single amplitude shear strain. We see large increases regardless of CSR in the number of cycles required to achieve half of the pore pressure generation as we increase cementation. And so these results were quite dramatic, again, showing that for the first half, the cementation had a huge effect on that first bit of pore pressure generation. As we then moved towards the second half, what we saw is really an increase, but a more modest increase. The slopes of these were starting to flatten out, and what we see is really the cycles from 0.475 to 0.95 were much less significant. That is, the cementation effect or benefits were diminished at higher excess pore water pressures. It really follows that it appears that the cementation initially is relatively stiff, and so for the first half of pore pressure generation, that effect is quite large. And as we're moving towards higher excess pore pressures, lower effective stresses, as we're damaging the cementation, we're seeing less of an effect. When we compare it now with the effect of densification, we can complete the exact same plots, still cycles for the first half and cycles for the second half on the left and right plots respectively. What we see is, again, densification does start to increase the number of cycles for the first half and the second half, but it's a really small effect when we compare it to cementation. We're still seeing much larger benefits from the cementation, both at the beginning, the first part of pre-triggering, and the second part of pre-triggering, regardless of densification whatsoever. We then looked a little bit further, and so now what we're plotting actually for a given test, these are single test results, we plotted the shear strain and the maximum RU felt by that specimen at that particular shear strain. And so what we were hoping to see here is maybe change in sort of the stress strain behavior, if you will, or the effect of pore pressure generation in these materials. What we ended up finding is that the more strongly bio-cemented samples, that would be your dark red here, when we compare it to the light pink here, which is our uncemented specimen, they can tolerate really high excess pore water pressures, or RU of something like 0.6, before they start to accumulate significant shear strains. Instead, for the uncemented specimens, really it doesn't take much of pore pressure generation before that material accumulates shear strain, and it does so in a much more gradual way, where our cemented specimens essentially generate excess pore pressure and don't start to yield until a higher RU value. Our uncemented specimens start to yield and start to strain even at those smaller RUs. And so what we see is really a change in what we would say is our threshold RU values. Something that is only 0.1 for an uncemented specimen becomes something more or less like 0.6 as we get to our higher levels of cementation, and it's really a spectrum. What it suggests is that our more strongly cemented materials can tolerate these higher excess pore pressures, but once they do, the strain starts to accumulate quite rapidly, in fact, more rapidly than it does in the uncemented material. We can also now look at post-triggering behaviors, and in this part, we're going to look at specifically cycles from 3 to 9% single amplitude shear strain. Really, this is after the triggering event. So triggering has occurred, and now we're looking at the next 6% shear strain. And it's again plotted versus delta Vs from cementation. What we can see is there's not much of a trend here. As we extend out to higher cementation levels, this line looks pretty flat. And so there's a minimal increase in the number of cycles required to go from 3 to 9 with increases in cementation, maybe something on the order of 3 to 4 cycles, but certainly not the 2 to 4 orders that we saw previously in the pre-triggering behaviors. And again, when we compare it now to the effect of densification, we can see that cementation overall, there's some jitter here, but there's not really much of a trend overall, relatively small effect. But densification does afford significant improvements in that post-triggering strain accumulation, where really cementation doesn't seem to be able to keep up with the effect of densification here. In terms of reconsolidation strains, this is after the shearing event, and we're able to plot similar things. This is reconsolidation strain on the Y, delta Vs on the X, and we're able to look at sort of the trend in strain that occurs post-shearing event. Again, this line is pretty darn flat. We maybe see some benefit from the higher levels of cementation, but not much. When we compare it in the same plots, now looking at reconsolidation strain versus total relative density, what we see is that cementation basically clusters at almost a constant reconsolidation strain level, but densification, again, is able to afford much better improvements in that type of behavior as we extend out to increases in relative density of approximately 50, right? We're able to see much more of a benefit there in terms of those behaviors. We can also plot here sort of our VS measurements, which were completed during the cementation process. So this is the increase in VS from cementation versus the change in shear wave velocity occurring from the shearing and reconsolidation event. We can plot things on a one-to-one line, which basically suggests that all of the improvement afforded by the cementation was actually destroyed during the shearing event. This is partially due to the fact that we sheared everything to 24% double amplitude shear strain, which is a relatively large shear strain. And so at this point in time, our materials are essentially uncemented, right? We've afforded or destroyed all of the bonding that we had from cementation because of this shearing and reconsolidation event. And it follows really that the post-triggering responses, it makes sense that those weren't all that different between the uncemented and bio-cemented specimens. As you'll remember from some of the previous plots, the way that things were plotted in terms of total relative density, the cemented trends didn't look all that different when we got to post-triggering behaviors. So in summary, for the engineering behavior aspect, of course, soil column testing has allowed for engineering improvements afforded by bio-cementation to be quantified, for us to develop effective process monitoring techniques, and for new treatment techniques to be developed. Recent DSS testing was really aimed at understanding sort of the boundary between uncemented and cemented conditions. And what it showed is that really light bio-cementation can have a huge effect on the liquefaction triggering resistance. We're talking about 2 to 4 orders of improvement in the number of cycles required to trigger liquefaction. That's huge news, particularly for the potential to reduce cost and environmental impact for this particular process. In terms of light bio-cementation, however, once we strain it, once we yield it, it really has minimal benefits in terms of post-triggering behaviors. And so, of course, this is where our denser soils really exhibit the more favorable responses, and the cementation really has limited benefit at those higher strains. Continued testing, of course, will help to improve our understanding of the behavior of these sands with the ultimate goal of enabling field scale deployment. And what that entails is really understanding better the effect of different stress states and cementation levels on both of these pre-triggering and post-triggering behaviors in order to understand what might be needed for a particular application. Okay, so that's it in terms of the engineering behavior part. Now we're going to talk a little bit about some process upscaling experiments which were completed, and in particular these meter scale column experiments. Just to kind of give you an overview of some of the past research which has been done on this, really collectively over the past 15 years, many researchers have been looking at applying MICP at field scale. This pilot testing has largely used augmented microorganisms for things like directional drilling or surficial applications. And in all cases, we've learned something about the process, both in terms of monitoring and verification. More recently, we've shown that meter scale testing can be used and can demonstrate that indigenous microorganisms can also complete the process. We've used these opportunities to develop effective sort of quality control, quality assurance, and process monitoring techniques In one case here, we're showing now shear wave velocity in both a stimulated, this would be indigenous microorganisms, and augmented microorganisms. And as you can see, these are 3 different wells, and they were injecting in a non-uniform manner intentionally. But the difference between the shear wave velocity distributions between the two aren't all that significant. And certainly shear wave velocity and calcite content correlated quite well. And so it's a non-destructive geophysical process that we can use at field scale to sort of verify where the cementation is and how to control that improvement zone, both in magnitude and extent. We also took advantage of these tests to develop things including not only shear wave velocity, but cone tip resistance. And so now we're plotting change in shear wave velocity relative to the initial condition versus calcite content. You can see again, that's roughly a linear slope like we saw with the soil columns. And what we see in terms of cone tip resistance, or QC, versus the uncemented condition is initially takes some threshold amount of calcite content, something like 2%, before we start to see measurable improvements in cone tip resistance. But by and large, it's able to pick up the higher levels of cementation and reflect that in terms of a cone tip resistance change. We're also able to use other types of techniques developed by other researchers, which really look at a small strain stiffness, G max, versus a cone tip resistance. So really a ratio of a small strain to a large strain behavior, plotted versus normalized cone tip resistance. And what we're able to do is sort of use these types of metrics to actually track changes in cementation. And these values are coming from the same tanks which we showed from before, basically suggesting that we can plot our uncemented materials here, and our cemented materials coming from MICP follow KG, or they follow these slopes and zones that one would expect for cemented material, regardless of the fact that we're doing it with MICP and not other natural processes. We can also look and assess and quantifying dynamic behaviors at field scale. This is largely done through centrifuge testing, which is being completed through the CVVG. Of course, what we're trying to do here is understand system level responses, not just how the element behaves, but how does a foundation perform, or how does a system behave? And so in these cases, we've been able to develop essentially triggering relationships, both in terms of cone tip resistance and cyclic demand, as well as shear wave velocity. And of course, these are moving us forward towards being able to use these types of processes for actual engineering applications. In terms of kind of the more recent challenges that we've been looking at, of course, there are still critical gaps and challenges to meet. And as we move from the laboratory setting towards this field scale implementation, there's a variety of ones that we really need to address. And so the first of which, of course, is the need to reduce costs and environmental impacts by optimizing our material usage. We have limited understanding, of course, of field implementation in terms of maximum treatment extent, effective soil heterogeneity, and the ability to control treatment uniformity. There's a need to address produced ammonium byproducts and the environmental impacts of that, so that when we leave a site, we can ensure that we're meeting sort of the environmental benefits and meeting those goals initially. And limited understanding really of the expected engineering behavior over some design life, right? What is the permanence? How does these materials degrade over time? And so really our recent research efforts in this meter scale testing is going to focus on 3 of these different barriers. The first of which is going to be to try to reduce materials and energy. This has been done through a variety of different element testing. The first of which showed that we can reduce urea by approximately 86% during stimulation. What we're showing here is a higher concentration in the red, a lower concentration in the blue. This is stimulation, that's cementation. And really what it suggests is we can reduce the urea during stimulation by quite a lot and still get full ureolysis or full reaction progression as we transition forward. And so there's a large cost savings and environmental benefit that can be had there. We can also reduce urea during cementation. That is, we can reduce urea and we're still able to get similar improvements in engineering properties overall, regardless of that. We can also improve treatment extent and cementation uniformity. And so in that case, what we're doing is looking at ways to sort of control the reactive transport conditions. What I'm plotting here is urea C over C naught. This is basically 0 to 100%. And this is elapsed time. So what we're looking at is essentially a single treatment. This is cementation 3 and cementation 6. And what we're looking at is urea degrading in time over a period of hours. And the different traces here reflect different columns which have different amounts of organic carbon or yeast extract. And what we can see is when we change the total cell densities by changing the concentration of organics that we supply, we can actually control the stimulated urolithic rate. We can make it relatively flat and slow, or we can speed it up by adding organics, which should, of course, result in implications for where the cementation would be formed and where the urea would be hydrolyzed. We can also look at sort of reactive transport modeling techniques to understand the interplay sort of between flow rate and treatment extent and magnitude. And so this is just one example of a given flow rate here applying at a slow urolithic rate versus a fast urolithic rate. We're going to get more uniformity because we're able to transfer or transmit those reactants to further distances if we have a slow reaction rate overall. And if we have a faster reaction rate, there's more localization of the precipitation closer to the injection location, and potentially less at further distances because the reactants are consumed closer towards the injection location. We've also looked at various techniques to remove ammonium byproducts, specifically looking at changes on ionic strength and pH. And we've done a variety of element tests to try to figure out what are the optimal techniques, or at least what are the most beneficial techniques that we have on hand in order to try to remove some of those ammonium byproducts. And of course, there's a need to demonstrate all of these things at practical scales. These are just results from small scale feasibility, and so that we really wanted to show this more at practical meter scales that would be used in more practical scenarios. So the meter scale column experiments were 5 3.7 meter long columns. They were treated using 1D flow in only one direction. They had pore pressure transducers, aqueous sampling ports, and shear wave velocity sensors along the column length. And there were 3 sands essentially treated with 3 different stimulation and cementation techniques. The objectives of this test were the following, the first of which was to evaluate the potential of microbial activity to control cementation distribution by sort of changing the interplay between reaction and transport rate. We were also trying to demonstrate successful MICP using solution with those large chemical reductions both during stimulation and cementation, as I mentioned before. And we were also trying to demonstrate ammonium byproduct removal. Finally, we were trying to compare the different treatment techniques and improvement successes across different material types. So this is what the actual columns look like. Of course, they're going to be treated in one direction. They had 4 bender element pairs, which you can see there in the blue, and 4 aqueous sampling ports, which are there in the magenta. And the idea was that they were treated once daily using a constant flow rate. We're talking about something like 1.5 pore volumes being replaced every day, and a relatively modest flow rate used to drive the treatment solutions through the column. 3 poorly graded natural soils were prepared at 60% relative density. They had D50s anywhere between about 0.5 to 1 millimeter, and they were from different depositional environments. This is what the overall setup looked like. We had a treatment tank where things would be mixed. We had a pump here, which was basically driving the influent of the treatment solution into a pressurized injection well. We measured the pressure here, and of course the outlet well was essentially just a free surface. And so during the process, we're taking samples at all these different locations, and we're measuring shear wave velocity at each of these different bender element locations. In terms of treatment protocols, these were the 3 different ones that we used. We had 2 different stimulation protocols. One was fast stimulation, so this is something with high organics intended to achieve a high reaction rate or a high urea degradation rate. We also had a slow stimulation. This had less organics, was intended to slow down the microorganisms and essentially have smaller cell densities. We also had one which received an augmentation protocol, which was essentially targeting the same rate as the fast stimulation one, but instead we were using lab cultured Sporosacina pasteurii microorganisms, which were injected immediately prior to cementation. As you can see, the timeline looked like as follows, where basically we have a period of almost 6 days or so where we have stimulation, after which we're transitioning to cementation, and in all columns we're applying 9 cementation treatments. In the fast stimulation, this was 9, which occurred in daily intervals. In the slow stimulation, that was 9 treatments, but the intervals then changed to 48-hour retention periods in order to allow for all of our reactants to degrade in that time period. And augmentation looked quite similar, with a need to re-augment at some point actually due to low reactivity overall. When we look at shear wave velocity, now we're looking at delta Vs versus distance from the injection. Again, we're going from 0 to 4 meters, so this is a quite significant distance, and we're plotting things all the way up to something like 1,500 meters per second. Just to kind of reflect and kind of put it into perspective with our DSS test, our DSS tests were delta VSs of 100, right? And so we're talking about 15-fold potentially greater cementation in these types of tests than what we did before. In general, when we're looking at these different traces here, we've got slow stim in the green, fast stim in the red, and augmentation. In slow stimulation, we see generally more uniform improvement. This gradient is a flatter slope than what we see in, let's say, the fast stim. We also see greater improvement at those really far injection distances. In the fast stimulation, however, we do see greater cementation at short injection distances, albeit there is less uniform improvement and a shorter extent of improvement. By the time we get to, let's say, 3 meters or so, or close to 3.5 meters, there's not a ton of improvement which is occurring at the end of the column. When we look at augmentation, we really see only improvements at that first shear wave velocity measurement point, and anything beyond 1.5 meters or 1 meter or so is really not experiencing any improvement whatsoever. In terms of calcite contents, we can see basically the same results. Now we're plotting calcite content versus distance from the injection location. You can see the contours have a little more data points because we're able to sort of dissect the columns in more discrete sections. But overall, the conclusions are the same. In the slow rate stimulation, we achieve something between 2 and 4% generally to the end of the column. In the fast rate stimulation, we get 6 to 7%, let's say in the beginning 1.5 meters, but a huge drop really occurring towards the end or the latter part of the column. Again, augmentation really only localized near the injection location, not much improvement whatsoever after we get past 1 meters. And really the reason for this and the augmentation is the sense that when we inject these cells, it's sort of like injecting colloids and not dissimilar to sort of clay filtration or clay caking on sort of an injection location. What we see is that these colloids get filtered out. And so the majority of the injected microbes, although we applied enough to fill the entire column, most of them were localized right towards the injection source and didn't really distribute evenly across the length of the column. In terms of permeability reductions, we also again had pore pressure measurements at each of our different aqueous points. What we were able to see is really insignificant changes in permeability for all of our columns, despite having quite large changes in shear wave velocity. Really what this suggested is we stopped at 9 treatments, but we really could have kept going in a lot of these different columns and continued injecting. Of course, as we do so, we're going to require more materials, more chemicals, and it may not make sense for the particular application, but really what this suggests is that the material remains pervious and open to additional treatments if needed. These are some images here just showing the destruction of some of these columns. Of course, we would break them apart and section them into different intervals to try to understand cementation distributions, both as a function of length and also as a function of sort of depth within the columns themselves. And really competent materials behaving much more like weak rock and able to pull out in cemented chunks like that. In addition, before really calling the test complete, we also wanted to demonstrate the byproduct removal component. And so in this part, what we did is we also applied injections in order to remove the produced ammonium byproducts after cementation. What we did is we applied 12 pore volumes of a high pH of about 9.5 or so, high ionic strength. I believe the concentration was something like 200 millimolar calcium. And so this solution with high pH and high calcium, the intention here is to actually remove some of the ammonium which may be sorbed to soil particle surfaces through cation exchange. And so we applied this to all of our treatment columns, and we took aqueous samples as well as VS measurements to track byproduct removal and also make sure that when we're injecting this, we're not changing cementation integrity significantly. When we did that, we were able to get various contours. Here we're showing total ammonium versus distance from the injection location. As you can see here, in terms of the contours, these are different time points. And both in the fast and slow rate stimulation, we had initial ammonium concentrations that were quite similar, about 500 millimolar. And as a function of treatment or rinsing, we were essentially able to get those concentrations down to somewhere closer towards 1 millimolar or on that order. Of course, there's a gradient here with lower concentrations near our injection source and increasing away from the wellhead due to dispersion. In addition, we also looked at sort of aqueous ammonium at various points in time. Now we're looking at sort of from the soil measurements themselves, both before and after rinsing. By and large, what we're talking about is 90% removal at all locations. And if you integrate those across, what we ended up calculating is something on the order of 98% cumulative removal of ammonium that was produced during the treatment process. We also looked at changes in shear wave velocity during cementation, or sorry, during the rinsing process. And so what we're showing here is delta Vs versus rinse injection time. And so this was our first, 12 pore volumes, which is injected, and then we had some residence period. What you can see is there's some scatter. It goes plus 25 to minus 25 meters per second, but there's really no consistent trends in terms of injection and its effect on cementation integrity. So we really feel confident that the process has minimal effects on the engineering properties of these materials. In summary, what we found is the meter scale testing demonstrated that we can reduce our chemical usage, that we can effectively stimulate indigenous microorganisms, that we can achieve cementation uniformity over distances that are practical for field scale applications, and we can achieve cementation levels that far exceed what we need for improvement of liquefaction behaviors, and we can quantify ammonium byproducts, manage them, and remove them. However, really, we're looking at alternative processes as well, which may offer some benefits there. And moving forward, reaction transport modeling is really going to leverage these experimental insights to both extrapolate and enable design of field implementations by utilizing some of the information that we get to develop or design treatment flow regimes, as well as understand the magnitude and extent of the improvement obtainable. Okay, so now I just want to briefly talk a little bit about some of the permanence testing that we've done, and then we'll finish from there. So really the motivation, of course, is that our treatment solutions and environmental factors can have significant effects on precipitate mineralogy and morphology, and that's been largely poorly understood. And so if we plot here urea to calcium ratio, these are all sort of urea to calcium ratios from treatment solutions versus year of the study, as well as concentration in terms of millimolar versus year of the study, we don't really see much conversion. Over 2 decades of research, we're probably just as far apart today as we were when we started these types of experiments. And so there's really a large range of urea to calcium ratios that researchers are using, anywhere between 1 to 1 to 100 to 1, concentrations anywhere from 25 millimolar to 3 millimolar. And overall, we're adding various different treatment additives, but there's no consensus in our best treatment practices. When we look at sort of the mineral synthesis process, of course, it makes sense that our treatment techniques in subsurface environmental conditions should alter the products that we make. And so things like groundwater and seawater ions, as well as solution pH and mineralogy, should all be important things that are going to affect the process. And they may have important implications for the life cycle performance of these materials in particular. And so it's easy to do these calculations, but really the presence of microorganisms makes it so that sometimes we can have things that are thermodynamically hard to predict. And so really there's a need for systematic understanding of the effects of very isolated variables, and a need for really guidance about where MICP can be stable and effective, and maybe where other technologies should be used, right? MICP isn't certainly gonna solve every problem, and so we need to figure out where the most benefits can be achieved. In terms of mineralogy, of course, we say that MICP is microbial-induced calcite precipitation. Sometimes it's referred to as carbonate precipitation, but by and large, we're thinking calcite. Of course, calcium carbonate can exist as many different polymorphs. One of them is calcite, but there's also vaterite, aragonite, amorphous calcium carbonate, and others. Here's just a few examples of scanning electron microscope images of the different morphologies. And as you can see here, there's different crystalline structures, different physical properties, and it only makes sense that these are going to have both engineering and geochemical implications. We're talking about potentially differences in tensile strength and dilatancy, as well as, of course, permanence and solubility of these minerals. When we look at mineral stability long-term, this is going to be governed by a variety of things. One of them is what's referred to as the solubility product, or the solubility of the mineral. It's going to depend on the chemical composition of the surrounding groundwater and its transport. It's also going to depend on dissolution rates, or essentially things controlled by mineral surface area. We can look at it and sort of compare differences just in solubility between these mineral polymorphs. And what we can see is just for calcium carbonate, there's a change of approximately 2 orders of magnitude in their solubility as we transition from something like calcite to amorphous calcite. Of course, we can use things like activity ratio diagrams where we're looking at activity of carbonate and calcium in a given solution and comparing it to the solubility of a mineral. And that can allow us to try to understand certain sites and certain locations where one would expect dissolution versus precipitation. And so really our questions are, which minerals are we forming? And can we enhance them in a way that we get predominantly calcite, which would have the lowest solubility and presumably the longest permanence? We did some batch experiments, which were looking at these systematic variables, again, looking specifically at physical, chemical, and mechanical properties, both through visual monitoring, reaction kinetics, and mineralogy, and relatively small batch experiments, which we can complete in the lab and are relatively small volume, so we can look at a variety of different treatment conditions. Of course, what we're looking at is mineralogy, morphology, and reaction kinetics as a function of all of these treatment factors or environmental variables, right? The environmental component we can't control, but we can certainly control what we inject in the ground in terms of our treatment solutions. And so the main ultimate question that we were trying to answer is, how do synthesis conditions alter and control the produced bio-cementation? And so this is overall kind of what our setup looked like. We had a plate batch reactor experiment. It contained bacteria, cementation solutions, and sand. The aqueous samples were obtained in time to understand precipitation kinetics and ureolysis. We did Xrd, FTIR, and SEM, both to understand precipitate morphology and mineralogy. And in some cases, we used biogeochemical modeling to try to inform our experiments and try to evaluate the effect of various factors. I would just want to share some results looking at the effect of urolytic rate and chemical concentration. Here, we're just changing the cell density, and here we're changing the chemical concentration magnitudes that we're applying. I'm not going to show you all the results from our chemical kinetics, but just focus on one aspect here, which is the Xrd analyses, which can tell us relative percentage of calcium carbonate as a function of different variables. And so on the left-hand side, we're showing you results from the different rate changes experiments. And on the right-hand side, these are the different concentration experiments. What we see is that generally we see for the rate changes a reduction in calcite and an increase in vaterite and aragonite as the microbial reaction rates increase. And with our concentration experiments, what we really see is at higher concentrations, we also see a reduction in calcite and an increase in vaterite, but not really a change in aragonite. Of course, these differences in composition are expected to have significant implications with respect to permanence and solubility. So if we can understand how these trends affect things, we might not inject at high concentrations and we might not use high rates. When we look at these materials in terms of Sem imaging, what we can see is that sort of at our standard concentration, 250 millimolar, get 95% calcite. Again, the morphologies are very cubic, look very characteristic of calcite here. As we move to higher concentrations, we start to see the appearance of spherical vaterite, really suggesting that the mineralogy is changing, and we pick that up in the Xrd, about 24% vaterite here. We can also look at a variety of other factors, including growth factors, spectator cells, and soil mineralogy. I just want to show you a couple results. I know we're heading towards the end of this here, so I'll make this part quick. We've got growth factors and spectator cells. With growth factors, we change concentration. With spectator cells, these are essentially non-urolytic cells that are just present during the precipitation process. What we see is with higher organic carbon, we get a reduction in calcite, an increase in vaterite and aragonite. In spectator cells, we see little changes in the mineralogy despite adding other cells into the mix. In terms of Sem imaging, again, growth factors are showing us the presence of vaterite in terms of the morphology. You could imagine if you try to shear something like this versus the cubic calcite from before, the engineering behavior would be very different, let alone the physical properties in terms of permanence. If we look at spectator cells, this doesn't even look remotely close to the stuff that we saw initially in our control experiments. In fact, we sort of see clusters of cementation reflective of essentially precipitation occurring over cell clusters. We also looked at changes in mineralogy. The long and short of it is we did see some changes on reaction rates, but we really didn't see any changes whatsoever on mineralogy. We're doing additional experiments to look at dissolution rates, of course, trying to be able to predict how quickly these things are going to degrade. And so answering the question, how long does bio-cementation last? What sites would be more favorable? And can we predict bio-cementation longevity? So we do dissolution batch experiments where we add in different acids and try to understand how the calcite is dissolving in time. This is feeding into numerical modeling efforts, which are looking at contours of calcium carbonate as a function of injected distance in something like a column like so. We've completed a recent experiment here, and I'll just show you one small bit of this, and that'll be the end of it. But essentially 5 different columns where we're injecting up to something like 50 dissolution injections, and we're monitoring it with BS, calcite content, and modeling. In the end, what we see is essentially that shear wave velocity and calcite content measurements can track these changes. Here we're injecting from the bottom and we're degrading the calcite in sort of a 1D type of flow regime. What we can see is the bottom is reducing in calcite, and we're able to pick that up during with BS measurements. At the top, we don't see much in a change of shear wave velocity, basically saying that all the dissolution is really localized to the periphery of the cemented zone. And we can track it both in terms of injection distance and calcite content, of course, the red correlating to our bottom shear wave velocity and the blue correlating to our top. I'm going to just skip this summary slide just for sake of time and move towards the final conclusions here. Okay, so in conclusion, MICP can be used to significantly improve the engineering properties of loose granular soils with potential environmental benefits relative to traditional ground improvement technologies. Our recent testing, both in the DSS and centrifuge and other venues, suggests that really significantly less cementation than we previously thought is needed to dramatically improve the resistance of loose soils to soil liquefaction. In terms of field-ready strategies, we've been developing those, and really we think we can offer large reductions in material and energy usage by optimizing the solution chemistry and using native microorganisms in the process. Shear wave velocity and cone penetration testing, of course, offer the ability to characterize MICP-treated zones, and there's always room for new verification processes there. And our meter scale experiments have shown that loose sands can be improved uniformly at distances exceeding 3 meters, and we can remove produced ammonium byproducts effectively. Just to kind of put this in maybe a broader perspective here, MICP, of course, is one tool in a larger suite of biogeotechnical solutions. And I think it's really exciting to think about how we can mitigate damage from natural hazards using these environmentally responsible and unconventional processes. And really, these are going to be used in conjunction with traditional processes, right? And so I think it offers a great opportunity here. There's also 1 million to 1 billion microorganisms that exist within a single gram of surface soil, and their extreme diversity and capabilities really offers an endless potential to develop new technologies beyond those which we've identified currently. And so I think this is really a field that's ripe for discovery, and it's a really exciting time to be a part of this community. With that, I'd like to thank all the students and faculty members that played a part in doing all of this fantastic work, and so it wouldn't be possible without them, as well as our funding agencies, including the National Science Foundation and the Center for Bio-mediated and Bio-inspired Geotechnics. And with that, I'd be so happy to answer any questions you may have, and thanks so much for letting me speak to you all and for giving me the opportunity to present. I've also included a QR code here, so if you scan that, that'll take us to our research website as well. All right, thank you so much.